Now, am I clear to everyone?
Yes, sir.
Yeah.
Yes, sir.
So right now, what you can see, guys, that is Cdisc. Cdisc is a non-profit organization. They create standards for clinical. So you can see here for organizing clinical data, they have a standard name SEND. For planning of clinical data, they have a standard name PRE. For collection, they have a standard name C-. For organizing, they have a standard, major standard is Sdtm. For analyzing, they have the standard name ADAM. Now, in our role, we should majorly know Sdtm and ADAM. And the very much important is Sdtm. C-, PRM, send. These work are done by different department, clinical data management, and before that, there are some other department, preclinical department. That is their work. In our work, because we need to create Sdtm and ADAM data set, so we should know their standards as well. You don't need to read the entire document, but you should have an overview of Sdtm and ADAM. So that's why first we wanna refer Sdtm IG, Sdtm Information Guide. So it tells us that what does it contain and what gonna be inside that. Yes, Sharon. Now share your screen.
Okay, sir.
So guys,
Yes, sir.
right now on the screen, the document which you are seeing, that is created by us. Okay. This is an PPT. And there is an official document as well from Sdtm, from Cdisc itself for Sdtm. Okay. So first we're gonna understand about Cdisc. Yeah, Sharon, continue.
Clinical Data Interchange Standards Consortium, Cdisc. Introduction of Cdisc Sdtm. Introduction. Cdisc stands for the Clinical Data Interchange Standards Consortium and is a global not-for-profit organization that actively develops data standards with the collective knowledge and experience of volunteers within the pharmaceutical industry. It creates and communicates standards that support the acquisition, exchange, submission, and archive of data for medical and biopharmaceutical product development. Cdisc standards cover everything from planning and data collection through to data analysis and reporting. Cdisc standards can be grouped into two different areas: content standards, data exchange standards. Yes, sir. Should I continue reading?
Yeah, let me explain the first para. So guys, in the first para, you can see Cdisc stand for Clinical Data Interchange Standard Consortium. This is the full form. So yes, you should know the full form. There is no something like urgency of this, remembering this, but yeah, this is an organization which creates standards for entire clinical data. Okay, so you should at least know the full form. Okay, and it is a global non-profit organization. Non-profit or not-for-profit organization is kind of NGO. They have registered their self NGO. Kind of they are saying we are not charging anything for these services, but obviously they'll be earning a lot from this. That actively develops data standards with the collective knowledge and experience of volunteers within the pharmaceutical industry. Why they have used the term volunteers, not employers? Because they are telling
Because it is a
themselves as a non-profit organization, an NGO. So an NGO cannot tell their people and employers, right? They are not paying for that. That's why volunteers. So who will be the volunteers here, guys? Managers, directors, seniors, experts. They might be contributing their expertise, right? Because this is for human welfare. It creates and communicates standards that support the acquisition. Acquisition means how to acquire clinical data, exchange, how to transfer clinical data, submission, how to submit clinical data, archive, how to draft clinical data for medical and biopharmaceutical product development. Cdisc standards cover everything from planning and data collection through, yeah, the same thing they are repeating. So we can move to the next slide, Sharon.
Cdisc content standards. Content standards. Plan, PRM, collect, C-, organize, Sdtm, send. Non-clinical, analyze, ADAM.
Now, these are some standards, content standard. Initially by opening the website, even after taking the screenshot, I have shown you, right? So this is the same thing, we have documented it. And for us, we should know Sdtm and ADAM. We're gonna read or we're gonna see those documents as well, the Sdtm and ADAM as well. Not ADAM, especially Sdtm.
Data exchange standards. ODM XML, operational data model, dataset XML, dataset data, define XML, dataset metadata, analysis results metadata, lab.
Mhm. Okay, go down.
History of Cdisc. 1997. Cdisc was founded by Dr. Rebecca Kush. 1999. Release of the first Cdisc standard, the Steady Data Tabulation Model (Sdtm). 2004. Cdisc recognized by the FDA for its role in regulatory submissions. 2014. The FDA mandated the use of Cdisc standards for regulatory submissions. And clinical standards. Sdtm, Steady Data Tabulation Model, defines a standard structure for clinical trial data tabulation datasets. ADAM, Analysis Data Model, specifies standards for analysis datasets to ensure traceability and reproducibility of analysis results. C-, Clinical Data Acquisition Standards Harmonization, provides standardized data collection instruments. Sir, should I continue?
Yeah.
Non-clinical standards. SEND, Standard for Exchange of Non-Clinical Data, ensures uniformity and consistency in non-clinical study data. Purpose, facilitates the review process and enhances data quality for non-clinical studies. Operational standards. ODM, Operational Data Model, facilitates the interchange and archiving of clinical trials data. Laboratory Data Model, ensures standardized structure for laboratory data in clinical trials. Detailed look at Sdtm. Structure, organizes data into domains such as demographics, adverse events, and lab results. Implementation, used to submit data to regulatory authorities like the FDA and EMA. Detailed look at ADAM. Traceability, ensures that all data transformations from raw data to analysis datasets are documented. Analysis ready datasets, designed for statistical analysis and easy review by regulatory agencies. Detailed look at C-, CRFs, case report forms, provides standard templates for CRFs to collect consistent data across studies. Compliance, ensures consistency in data collection across different trials and organizations. Other Cdisc standards. Controlled terminology. Cdisc provides controlled terminologies and code lists for consistent data representation. Data exchange standards. Cdisc has developed standards for data exchange formats such as the Data XML SDX standard. Therapeutic area standards. Cdisc has developed standards for specific therapeutic areas such as cardiovascular, diabetes, and oncology. Cdisc and regulatory submissions. FDA requirements. Since 2016, the FDA has required CDSC standards for electronic submissions, global acceptance, increasing adoption by regulatory agencies worldwide, including EMA and PMDA. Benefits of using CDISC standards: improved data quality and consistency, streamlined data exchange and submission processes, exchange data traceability and transparency, facilitated data review and analysis, increased operational efficiency and cost savings, standardized representation of medical device data. Thank you.
So in short, guys, Cdisc as a non-profit organization, they create standards for clinical data. And even you can read the standards name they create. But for us, the important one is, as per our role is Sdtm and Adam. And the very important is Sdtm. Now we're gonna have a look on Sdtm. So in the docs folder, open the Sdtm PPT.
In the doc,
Yeah. There is no Sdtm.
no, sir.
Refresh it. Refresh the folder.
Yes, sir. Is this SDTMIG? First one. Or
This
this?
is official. Yeah. Official one we'll see it later. First, we'll
Okay.
see the PPT we have created. So Sdtm,
Okay.
that IGP
Second one.
PDF is, yeah.
Mm.
Now, guys, what is Sdtm IG? That is one of the document created by Cdisc, okay, to standardize the clinical data. Instead of I'm explaining this, let us understand one by one, point wise.
Okay. Cdisc, SDTM, and SDTM-IG. Introduction. Sir, say the.
Yeah, just read it, no worries.
Okay, introduction. Cdisc stands for the Clinical Data Interchange Standards and Consortium and is a global not-for-profit organization that actively develops data standards with the collective knowledge and experience of volunteers within the pharmaceutical industry. It creates and communicates standards that support the acquisition, exchange, submission, and archive of data for medical and biopharmaceutical product development. Cdisc standards cover everything from planning and data collection through to data analysis and reporting. Cdisc standards can be grouped into two different areas: content standards, data exchange standards. Cdisc content standards: plan, PRM, collect, organize, SDTM, send, non-clinical, analyze, ADAM. Data exchange standards: ODM XML, operational data model, dataset XML, dataset data, define XML, dataset metadata, analysis results metadata, lab. Introduction of Cdisc SDTM. The steady data tabulation model is one of the most important Cdisc data standards that is used for organizing data collected in human clinical trials. It gives regulatory reviewers a clear description of the structure, attributes, and contents of each dataset and the variables submitted as part of your clinical trial, where the SDTM provides a standard model for organizing and formatting data for human and animal studies. The SDTM IG is intended to guide the organization structure and format of standard clinical trial tabulation datasets. Sir, should I continue?
Yes, Sharon, just allow me for a minute. So guys, now we are seeing one of the document created by Cdisc. The document name is Sdtm. The full form is study data tabulation model. How to tabulate the study data, how to standardize the study data. For that, this document provides a model. Is one of the most important Cdisc data standards that is used for organizing data collection in human clinical trials. It gives regulatory reviewers a clear description of the structure, attributes, and contents of each data set and the variable submitted as a part of your clinical trial. Where the Sdtm provide a standard model for organizing and formatting data for human and animal study, the Sdtm IG is intended to guide the organization, structure, and the format of standard clinical trial tabulation dataset. In short, what they are telling, the name itself contains the whole story. Sdtm, Study Data Tabulation Model. So the data we are collecting for this study, how to tabulate it, how to standardize it. Put on.
Sdtm domains. A collection of logically related observations with a common specific topic that are normally collected for all subjects in a clinical investigation. Special purpose domains, general observation classes, findings, trial designs, relationship datasets. So under special purpose designs, that is demographics, comments, and findings, that is findings about skin response, trial designs, that is a trial summary, trial arms, and trial visits, relationship datasets, that is a supplemental qualifiers. Under general observation classes, there are three subdivisions: interventions, that is exposure, substance use; events, adverse events; disposition; findings, that is vital signs, physical exam. Special purpose domain datasets can be demographics, comments, subject elements, and subject visits. Demographics. A set of essential standard variables that describe each subject in a clinical study. Key variables are study ID, uh, U subject JID, U sub JID. Subject elements contains the actual order of elements followed by the subject together with the start date or time and end date or time for each element. Key variables are study ID, U sub JID, ETC, SES, TDTC.
Guys, so right now what you are seeing, these all are data sets. Patient data are collected in a form. We have seen, right? CRF form, case report form. And after that, from that form, it will be converted into a SAS data set by Cdm team. It will be collected in a data set. Not all the data will go to a single data set. The data will be segregated or separated into multiple data, the subject data. Now, who have decided that this data will go to this data set, this data will go to this data set? For that only we have these standards. They have already mentioned that the study data or the patient data will go to which different data set. Like a same patient name, sex, age, height, weight data can go to demographic. And their visit data can go to a different data set named subject. That is predefined. Same way you can see demographics, DM, it contain subject ID or start date or end date, age, sex values. Then the second data set subject element, it contain the data like, few information will be common in each of the data set. Study and ID and subject ID, because these are identifier variables. This is how you can identify, right? Whose data is this? Which subject data is this? But what does subject element contain? It contains the element followed by subject. Element means subject is in which part of the clinical trial, like follow-up phase, screening. You can see there is a variable name element. It shows follow-up, and the next it shows screening. Screening means as of now, subject screening is going on. Follow-up means subject treatment is completed. He is in the follow-up phase. And then start date and date, each and every information is captured. Same way in different data set, different values will be kept. Different data will be collected. Go down.
Subject visits contains the actual start and end date or time for each subject for each visit of each individual subject. Key variables are study ID, use subject ID, visit number. Yes.
Go down.
Trial designs. Trial designs has datasets that describe the design of a trial. Examples are trial summary, trial arms, and trial visits. Trial arms. This describe the planned design of the study and provide the representation of study treatment. Key variables are study ID, arm CD, TAETORD. Trial elements contains the element code that is unique for each element, the element description, and the rules for starting and ending an element. Key variables are study ID, ETCD. Trial visits contains the planned order and number of visits in study within each arm. Key variables are study ID, arm, visit. Interventions datasets capture treatments and procedures that are given to a subject as specified by the protocol. Examples are exposure, concomitant medications, and substance use. Example tobacco, caffeine, and alcohol. Exposure. It contains the details of a subject's exposure to protocol specified study treatment. Key variables are study ID, use subject ID, EXTRT, EXSTDTC. Concomitant medications. It contains concomitant and prior medications used by the subject, such as those given on an as needed basics or condition appropriate medications. Key variables are study ID, use subject ID, CMTRT, CMS, TDTC. Substance use. It contains substance use information that may be used to assess the efficacy and or safety of therapies that look to mitigate the effects of chronic substance use. Key variables are study ID, use subject ID, SUT, RT, SUS, TDTC.
So
Even
what we can do here, instead of you are reading all those things, I can just give a brief about this data set. So guys, these things you will understand once you create the real data set. But I'll give you a brief. What are those names? Like what data it contains? Concomitant medication. If the patient who are enrolled in the study, and if during the study, if they are taking any other drug apart from the study drug, let's suppose one drug is study drug that is being tested on them, right? Another drug can be, let's suppose due to study drug, patients start feeling the headache. And for headache, there are some common drug available in the market, right? I think what are the names, Sharon? Dolo, or maybe these medicines are there, right? Some
yes,
for headache. So whatever any other medication they are taking or the doctors are giving to them for any any problem they are facing, not problem, we can say.
sir, diabetes medicines and hypertension medicine. Few people will take Nasir for sugar, BP.
That can be also considered. So any other drug apart from the study drug, any other drug they are taking due to sudden event, they are feeling chest pain, head pain, or existing drug they were taking since long, like diabetic or any other drug they were taking, that will be, those data will be collected in concomitant medication data set. Okay. And the dose unit, you can see CMD code. CMD code, you can see the coded name, and CM dose TXT. In that you can see the dose amount, 300 mg, 1000 mg. What is the, that is mg is the, power, I think, of the drug or unit. And then the next data set is substance use. Substance use means, let's suppose we are enrolling patient in the study. Okay. So there, patient can be consuming in the past drugs, tobacco, alcohol, anything. Maybe right now they have left, or they might be consuming right now as well. So, whenever we are enrolling any patient, at the time of screening, we have some criteria. Along with the criteria, we collect a lot of data of the patient. Like, let's suppose from past 5 years, if they were consuming, in past 10 years, if they are consuming drug, alcohol, tobacco, even coffee, if they are active, they are actively preferring chai or coffee, anything. It also contains caffeine, right? So we collect each of this data, because if a patient is having, if the patient was consuming a lot of alcohol in the past or currently as well, so it can affect our study, right? There are much more chances that there can be adverse event, or there can be side effect on this particular group of people, because their immune will be, weak, right? The patient who are already consuming a lot of bad things in the past, or they have already gone through, some severe problems, so the chances are high. So substance use, we will be collecting the data. What are the substance they were consuming in the past or in the present? It can be tobacco, it can be alcohol, it can be drug, it can be coffee, it can be anything. So once you'll try to understand this data set, now you'll get better clarity, instead of, right now we are reading because we are seeing this for the first time. And we will not only read, we gonna create the data set. Yeah. Go down. Adverse event. This is also very interesting. It contains the data describing untoward medical occurrence in the patient. Let's suppose during the study, due to study drug, or due to any other reason, patient started feeling shivering or head pain. Anything happened to the patient, that will be counted as an adverse event. Some event happened with the patient. Adverse event. Adverse means something wrong, right? Something which is not good. So during the study, if any event happened, so that will be recorded. That data will be recorded in this data set. So you can see the first variable, AE term. Can everyone see the first variable, AE term? So here, what is the term? What's the reason for adverse event? The first one.
Injection site reaction.
A patient, and what was the event? AE, BOD, CYS, read in that variable. Third variable.
General disorders and administration sit mild.
Yeah. So due to injection reaction, something happened to the patient. Okay, that term is mentioned over here. So, like, what happened, or due to what event occurred, this will be recorded. And what is the severity of event? Mild. And what was the start date of event? The last variable is mentioned, AE Sdtc. This is how all the data we are gathering in different data set. Go down. Okay. Disposition. Disposition is means, yeah, go ahead.
Disposition. An events domain that contains information encompassing and representing data related to subject disposition. Key variables are study ID, subject ID, DSDE, COD, DSST, DTC. Medical
Disposition, yeah, disposition can contain data. Let's suppose, I'm not sure, I don't remember exactly, but let's suppose if a subject is leaving the study. So let's suppose here I'm assuming DS term is mentioned subject eligibility. So it means maybe this subject have failed in the screening. During the screening, we have some criteria, right? Uh, what are those criteria, guys?
inclusion and
We have
exclusion criteria.
inclusion and exclusion criteria, right? So these subject might failed, in that criteria. There can be other reasons as well. Subject is no more. There was some adverse event. It can also happen. There is some adverse event due to that. Subject is no more, and due to that he's no, he's leave. Not he is leaving now. He'll be not continuing the study. Then the next data set is medical history. The name itself is telling what data it's gonna contain, guys. So what data it's gonna contain? Medical history. It means, let's suppose in the past 5 or 10 years, patient have gone through some surgery, patient have gone through some, accident, anything, any information related to their medical, that will be captured in the medical history data set.
Findings data sets. Capture observations that address specific questions such as observations made during physical examinations, laboratory tests, ECG testing, etc. Examples are vital signs, inclusion or exclusion criteria, not met, and lapse. Vital signs. It contains measurements including but not limited to blood pressure, temperature, respiration, body surface area, body mass index, height, and weight. Key variables are subject ID, study ID, subject ID, VS test CD, visit number, VSTP TREF, VSTP T number.
So guys, this is related to vital signs data, that is related to body, respiration, like mass index, height, weight. This data will be captured in vital sign data set.
Inclusion or exclusion criteria not met. It contains those criteria that cause the subject to be in violation of the inclusion or exclusion criteria. Key variables are study ID, subject ID, IE test CD. Labs.
So,
It contains
so guys, this data set inclusion, exclusion, IE, by the name itself, you can understand what data it gonna include. So let's suppose if a subject got excluded from the study due to any other reason, or he got included or excluded, that data will be recorded in this data set. You can see IE test. IE test contain what test he gone through, and it was an inclusion criteria or exclusion. So type of participant and disease characterized in good journal health and evidence. Stated willingness to comply with all study procedures and ability for duration inclusions. Then lab
lab.
data. So Sharon, we can leave this. No need of reading all those things. Go down. Yeah,
SD,
this you can continue.
SDTM variable roles. Each domain has a dataset which is a collection of related data. SDTM datasets are described by a set of named variables, and each of these named variables is categorized by their role. A role category conveys a particular type of information about a variable. Variables can have just one role.
Yeah, continue.
Variables. Identifier identifies the study, subject, domain, and sequence number of a record. That is study ID, use subject ID. Topic describes the focus of an observation, that is test CD term. Timing describes the date, time and duration of an observation, that is STD, TC, DTC, DY. Qualifier describes the results of an observation with text or numeric values. Rule describes algorithms or methods for calculations or looping conditions mainly in the trial designs domain, that is TATRANS ENRL. SDTM core variables. Core variables are a measure of compliance with the specific Sdtm IG domain model. The value of a core variable shows the importance of the variable to the overall domain structure. The concept of core variables in the Cdisc is to control variables with missing values in the submissions. Variables are divided into three categories. Required variables. Required variables are needed to identify a data record. Example, study, study ID and a use subject ID, or they are needed to make a record easily understood. Example, term and test. They must always be included in the dataset and cannot be null. Expected variables. Expected variables are needed to make a record useful within a specific domain. They must always be included in the dataset, but they can be null for some records. If no data is collected, a comment must be included to explain why. Permissible variables. Permissible variables must be included in the dataset if results are collected or derived, but they can be left null or blank. Thank you.
So guys, this, you have to read little bit. You don't need to understand each and everything, but if you read, what I feel that the much more time you spend in reading the SDTM IG, the much more clinical related knowledge you're gonna get. Okay, but actually you're gonna do practical. So while doing it, because your work is not to read this document, your work is to create data set. But yeah, these are the contributor of the output you're gonna generate. So if you read this, so you can understand or relate more what I'm doing and why I'm doing. Okay. So there are two ways of understanding anything. One way is to gather enough information before proceeding. Another way is to do the practical. From the beginning itself, we have, in everything we have done practical and we try to understand by that because we feel that doing it is a better way of understanding instead of reading it. Now, this is the PPT we have created. Now we're going to see the SDTM IG official document created by
Cdisc.
Cdisc. Close this document. Yes. Now, guys, the document we are seeing, this is the official document created by Cdisc. You don't need to read the whole document. I'll tell you what are the things you can read. Scroll down. Yeah, here, fundamentals of SDTM. Go to the first, read the first para, Sharon.
Fundamentals of the SDTM. Observations and variables. The v3.x submission data standards are based on the SDTM's general framework for organizing clinical trials information that is to be submitted to the FDA. The SDTM is built around the concept of observations collected about subjects who participated in a clinical study. Each observation can be described by a series of variables corresponding to a row in a data set or table. Each variable can be classified according to its role. A role determines the type of information conveyed by the variable. about each distinct observation and how it can be used. Variables can be classified into 5 major roles. Identifier variables. Identifier variables such as those that identify the study, subject, domain, and sequence number of the record. Topic variables. Topic variables which specify the focus of the observation, such as the name of the lab test. Timing variables. Timing variables which describe the timing of the observation, such as start date and end date. Qualifier variables. Qualifier variables which include additional illustrative text or numeric variables that describe the results or additional traits of the observation, such as units or descriptive adjectives. Rule, rule variables. Rule variables which express an algorithm or a executable method to define start, end, and branching or loop
Yeah,
conditions.
this much is fine.
Trial design.
Yeah, let us try to understand, guys, the first para. Submission data standard are based on Sdtm journal framework for organizing clinical trial information that is to be submitted to FDA. The Sdtm is built around the concept of observations collected about the subject who participated in clinical study. This Sdtm is built around the concept of observation. See, what what we are trying to do, we are trying to standardize the data. What is data? Data is nothing but observations. Observations, right? In a variable, we store the observation. That is the important. Each observation can be described by a series of variables, like the observation where it contains names. So it will be stored in which variable? Name. The observation which contain sex related data, it will go to the gender. The observation which contain age related data, it will go to the age variable. So here the same thing they are explaining in different way. Each observation can be described by series of variable corresponding to a row in a data set or table. Each variable can be classified according to its role. A role determines the type of information conveyed by the variable. It's very simple. The variable name defines or determines what data it's gonna contain. Now go to page, okay, go down. Yeah, here it is, page number 14. Now, this is very important. Now you need to try to, memorize this thing. Until unless I never told you to memorize anything, but this thing is very important. You are seeing the names, right? Comment, subject, element, demographics, subject visit. These are nothing but data set name. The study data we are collecting, that is going to be stored in these data sets only. By reading some of the names, you can easily understand what data it's gonna contain. Subject visit. Subject visit data gonna contain subject visit related data, right? Adverse event. It gonna contain if any adverse event happened with the patient, that data. So these are the data set name. There is a standard we follow. Data set name will be with 2 words only, CO, but the full name is also mentioned. Like demographics will be known as DM. Subject visit will be known as SV. So there are total how many domains? I think 7 domains. Domains are, the name within that data set is stored. Special purpose domain.
7 is there.
Yeah. Intervention journal observation class, and in that data set name is there. So at least try to remember 2-3 data sets from each of the domain. Guys, even if you don't remember, nothing to worry about, but if you remember, you can relate little bit while creating it. Because these are the data sets we're gonna create. Now this is the official document, guys. Whenever you get free time, spend time in this document, because this is the documents we are following in each and every clinical trial. No matter which company you're gonna work, no matter you're working in US or India or Canada or Europe, everywhere the same standard gonna be followed. This is a global standard. Whenever you work on a clinical role or clinical SAS programmer role, that is a global role. You are not working for one country. Your work is global because you are following a global standard. This is a global standard. So spending time on this document is valued. And software part you have already learned together, because let's suppose in your academic these things are not taught. So this information, if you want to ensure I'm at the top level of clinical, even in the interview, even while working, reading this document will worth it. But as a fresher, reading a lot this document, if you're not interested, that is not useful. Because if I'll tell you, when I got a job, even I haven't seen this document. I was not knowing about this document. Okay, nothing about Sdtm. Nothing like, yeah, I have just read it, the definition, that's all. I was knowing some part. So yeah, but it gives you the clarity behind the scene understanding. So what is your work now? You have to read the Cdisc IG. Cdisc IG is like very easy. They are the creator of standards like clinical data and interchange consortium. That is a full form. They create standards for clinical data. But the very important is this Sdtm IG document. Try to memorize this dataset's name and domain's name and read the important parts of this Sdtm IG. You can read the PPT we have created. You can read this as well. And on top of that, go down. Actually, they have explained about the data set because they have created the standards. So that information is on simply in the search bar, simply write substance use.
Where search bar, sir?
You can see search option is there in this PDF at the right hand side top. Third bar. Yeah, here in this bar only search. Magnifying glass is there. No, not that one. Before that. The same line before that.
This.
No. Before that. Before. More. Go more.
Okay.
Yeah, first.
What should I say?
Substance, SUBSTANCE. Use. SU data set. I want to have a look.
Substance use.
Nothing, you're not getting anything.
No. Now how to execute this, sir?
It will automatically search. I'm sharing my screen. No worries, guys.
Okay, we got it, sir.
Got it.
Int.
I'll share you the better way, guys. Is my screen is visible to everyone?
Yes, sir.
I have opened the document. So let's suppose it's all about data set, right? So let's suppose if I have to open any data set, so you can see in the beginning contents is mentioned, right? So first you can read some of the things, purpose of Sdtm IGs, few things, fundamental of Sdtm, and then you can see the data sets name are there along with the intervention domain content these data sets. Exposure, substance use data is here. I can directly click here. It will directly go to that page number. Okay. So substance use data set, what we have discussed. So you can see here, these are the variable name it's gonna contain. And how the programming should be done, here it is mentioned. The variable name, variable label, variable type, control term, role, and Cdisc know how to create this variable. And core justifies it is a required variable, permissible variable. So I'll show you now these are the variable, but how it will look like after creation. Here it is. See, substance SUTRT. Now this is interesting. My screen is visible to everyone, right?
Yes, sir.
So what does substance use data contain? The substance used by the patient either in the past, either in the present. You can see SUTRT is one of the important variable which contain what substance this patient were using. So subject ID 1234055 was using cigarette. That comes under which category? Tobacco category. How many dose he will take? 2. And substance dose unit pack. It's not 2 cigarettes. He was having 2 pack of cigarettes. What is the frequency per day? Now, the same subject, the same 1234055, I think, he was taking coffee as well. That comes under caffeine category. And how many? 3 cup per day. Then again, 1234666. He was taking tea that comes under caffeine and 1 cup per day. You can see all the data. And then when did he started consuming cigarette or whatever substance he's consuming? What is the end date? All this data is getting collected. So reading about the data set in the same way, you can just read, just see the data. See, by seeing this data SU, you should understand, first read it from here. You can see here also it is mentioned which are the important variables. SUTRT in the definition, SUCAT. So by seeing itself, you should know these are the common variables. Study ID will be in each of the data set. Domain contain the substance use, data name, short name. Use subject ID will be in each of the data set. Sequence means this subject is data is collected second time. You can see the same subject is 2 times. One time we have captured his cigarette data and one time coffee. As this is something important, variable for this data set. UTRT, which contain the substance used by the patient. And then these are also important. The very much important variable will be this one, which contain the names and then the frequency, start date, all this. So understanding your data set by this way is, it will change the way you are looking. Now let's have an adverse event, look on adverse event data set. Adverse event may be captured either as a free text or pre-specified list of terms. It means the data related to adverse event. So these are the variables that gonna be in the adverse event data set. Now how it looks like, let's see close information. Will they have given the data set? Yeah, you can see. Now, which is by seeing the adverse event data set, what do you think, which will be the main variable? You have to read, yeah, for sure, little bit definition on those things. But what can be the main, important variables? Can someone tell? These are the variable names you can see on the screen.
Sixth variable.
First, second, third, fourth, fifth. Which one? Fifth or sixth?
Fifth.
First, fifth. A term, very correct. It contained what was the term or reason for adverse event? Pounding headache for one subject. Another subject, back pain for 6 hours. Third subject, embolism.
Pulmonary
And then,
embolism.
yeah, then AE start date, AE end date. When did this event started? Whatever adverse event. And what was the end? time or date. You can see headache, back pain, and pulmonary embolism. And AE body system, like related to which system organ this is. So nervous system disorder, vascular disorder, musculoskeletal and connective tissue disorder. What is the severity? So severity of this event. So it was severe. It was moderate for this subject. For this subject also moderate. So you can see if you try to understand your data in this way, so you can relate more. So what we are doing actually, we are collecting the patient data and then we're gonna analyze as well. And these are the standard name. When the data is available in the form, they are not available with these names. The name are different. As we have seen in the annotated CRF, right? Did I shown you annotated CRF? In the form, actually, simple names are given. And then after collecting it, we will give the standard name. Clinical events. So you see, this is how the data looks like in form, but this is how they collect it. See, clinical signs, rash, edema, and these terms are there. Then start dates are there. But how it got collected in data set? It got stored in a variable name CE term. But the simple name in the form will be clinical signs. But once we'll move to a data set, it will be stored in a variable name CE term. So this is how from a form is data shifted to this variables. But moving and the shifting things, we don't do it. There is a department, CDM, clinical data management. They take care of these things. We just need to do standardization. And what we do, you don't need to worry, guys, because in tomorrow's session, we're gonna create Sdtm. We are just trying to understand behind the scene. This is how the form look like. In the event, it is showing diarrhea or what. And you can see the CE term contain the reason for clinical event, vomit, diarrhea, severe head pain. So likewise, seeing the data set, if you just look the data set, if you just try to read it little, it will give you better clarity. So spend some time reading about the data set the way we have seen for some of the data set. It will give you the clarity because you want to create these variables only in real time. And these variables you want to create lifetime. It is not like for one study. Lifetime you have to create these variables. The only thing will be studies will be different, but the variable name is going to be same because these are standards. So whenever clinical event data is collected, it will go into which data set? Clinical CE data set, and the data will go in which variable in that data set? CETERM. Is this clear to everyone? Now,
Yes, sir.
guys, this is a very lengthy process, like understanding everything, the documents. It's not a one-day thing. It's gonna take time, but let's try implementing from tomorrow, so you're gonna get better idea. So guys, that's all for today. I have uploaded the required documents in the docs folder. Just have a look. Just have a look on Cdisc and then Sdtm IG and Sdtm IG official. Any questions, anyone, as of now?
No, sir.
That's all for today, guys. Thank you, everyone, and bye-bye, everyone.
Bye.
Thank you, sir.